Hope all of you are able to see my screen.
Yes, sir. Yes, sir.
So we are discussing, MPN test, so multiple tube fermentation technique for coliform bacteria. Now, before going into the details of MPN test, I would like to mention few things about MPN test. Usually, three type of MPN tests are performed. One is presumptive MPN test, and then we have one confirmed coliform MPN test, and then we have fecal coliform MPN test. So usually these three type of coliform these three type of MPN tests are there. However, in this particular course, we are going to discuss about the confirmed coliform MPN test. So I'll discuss them step by step. Just wait 1 second. Someone is waiting. Yeah. So, in this particular process, this is basically multiple tube method, and a series of tubes containing a suitable selective broth culture medium. So that a broth can be of different type, mostly lactose containing broth, such as McConkey broth is used usually. However, doing the proper bacteriological characterization, you can decide that which are the broths which would be suitable for this particular test. So, broth culture medium is inoculated with the test portions of a water sample after a specified incubation time at a particular given temperature. Each tube showing gas formation is regarded as the presumptive positive, since the gas indicates the possible presence of coliform bacteria. However, the gas may also be produced by other organisms, and so a subsequent confirmatory test is essential. And the two tests are known respectively as the presumptive and the confirmatory test, which actually I mentioned. Now, in case of confirmatory test, a more selective culture medium is inoculated. Fine. So, namely, brilliant green bile broth, which is usually used, inoculated with material taken from the positive tubes. So, in the presumptive test, in whichever tubes we have got positive results, from there the material is taken, and then again it will be cultured in selective broth medium. After an appropriate incubation time, the tubes are examined for gas formation as before, which we have seen in case of presumptive test. The most probable number of bacteria present can be estimated from the number of tubes inoculated and the number of positive tubes obtained in the confirmatory test. So, using specially devised statistical tables, this MPN most probable number can be obtained, and this technique is known as the MPN method. Fine. So here, what we are going to see is, the entire diagram of the, how we perform the presumptive coliform MPN test and confirmed coliform MPN test and the fecal coliform test in water quality monitoring. So we start from, surface water sample which we collect, then we inoculate with the lactose broth or l'oreal broth, and the temperature is kept as 35 degrees centigrade, and for the incubation period the time which is taken is 24 hours. Now there are two possibilities. We have no gas produced in incubator, so, and another one is gas produced. So for no gas produced, we'll again incubate it for additional 24 hours. For gas produced, we'll then divide them. One is we'll again, what we need to do is, now we have got the positive in the presumptive test. Now we'll have to go for the confirmatory test. So there are two different broth by which we'll do the test. So one is the brilliant green lactose bile broth, so which I mentioned already. Another one is EC medium broth. So we'll see again in the no gas produced category. So after 24 hours, if there is no gas produced, that means that is negative in the coliform, negative test for coliform. So that means there is no coliform group present in that particular samples. If we get gas which has got produced after additional 24 hours, then what we will do is we'll take those samples and then again we'll add the brilliant green lactose bile broth and we'll incubate it for 48 hours at 35 degrees centigrade. So then what we'll get, there will be two different possibilities again. One, no gas produced, so that will be again considered as negative test for coliform group presence. So there is no coliform group present in that particular samples. If gas produced, presence of coliform group confirmed, since we are doing now confirmatory test, and that is what is mentioned over here. If you see, so this is the gas produced presence of the coliform group. On the other hand, so once we have got gas produced in the presumptive test, one particular portion we have actually mixed with the same broth, and then the rest of the part is same. If we get gas produced, then it will be coliform group confirmed, and then if we see that there is no gas which has got produced, then we'll say that there is no coliform present in that. On the other hand, once gas is produced, we can do, in both the cases, we can do EC medium broth and we can incubate it for an additional 24 hours at 44.5 degrees centigrade at elevated temperature. So in this case, what we would be able to see is, if there is no gas produced, that means again no fecal coliform present, and if the gas has been produced, that means fecal coliform group is present. So this is the overall process from where we are taking the sample and then we are performing the presumptive test. At the same time, we are performing the confirmatory test for the coliform group presence or absent, and at the same time we are checking the fecal coliform presence or absence in the same sample. So this particular flow diagram is very much important, and you should study this minutely to keep these things in your mind. Fine. So now we'll be talking about the thermotolerant coliform. So through this process, we would be able to see whether coliform are fecal coliforms are present or not. So this particular process now I would discuss in detail. So presumptive have been discussed. Now confirmatory have been discussed. Now we are talking about membrane filtration method for total coliform and thermotolerant fecal coliform. So that is what we'll be discussing now. So the method is based on the filtration of a known volume of water through a membrane filter consisting of cellulose compound with a uniform pore diameter of 0.45 or 0.2 micron. The bacteria are retained on the surface of the membrane filter. When the membrane containing the bacteria is incubated in a sterile container at an appropriate temperature with a selective differential culture medium, characteristics colonies of coliforms or thermotolerant coliforms develop. That can be counted directly. Fine. In contrast to the multiple tube method, in the membrane filtration method, we get directly we get a chance to directly count the total coliforms and the thermotolerant coliforms present in that particular sample. So that is the main difference, if we talk about with the multi-tube MPN test which we performed before. And since the filtration area is relatively small, it can support the growth of only a limited number of colonies, and the optimum number is between 20 to 80 and with a maximum limit of 200. So the maximum is 200, so it can be countable easily. If this figure is exceeded, very small atypical colonies or superimposed colonies may develop, or there may be growth inhibition due to the overpopulation. Fine. So the choice of the volume of sample to be filtered will depend on the type of water. So that is why it is very important to keep this in the mind, that if there is an overpopulation, or if there is not distinct colonies which can be found, then it is very difficult to determine the MPN finally by counting directly. So that is why we have to take care of these factors, so that we get a value between 20 to 80 mostly, which is very convenient for us to count the overall colony and to obtain the MPN in the end. Volumes less than 10 ml should be added to the filtration apparatus after addition of at least 10 ml of sterile diluent to ensure adequate dispersal across the surface of the membrane filter. So if we follow that, the chance is higher to get colonies between this 20 to 80, and we will be able to count that. So the colonies of coliform for thermotolerant coliform bacteria should be identified from the characteristics of the medium used. So the number of coliforms per thermotolerant coliforms, per 100 ml is given by coliform. Per thermotolerant coliforms per 100 ml equals to within bracket number of coliforms by thermotolerant coliform colonies counted whole by number of ml of samples filtered into 100. So that is the way we would be able to determine the distinct number of fecal coliform present in a particular water sample. Fine. So there are several more things. I am not going in those details. Those are typical biological aspects. We'll now move to the next part. So that is the coliform index. This is very important, coliform index. So coliform index is a term which was lately used to measure the coliform bacteria present in water sample. So this is also one way to identify how much coliform bacteria are present in water. It may be defined as the reciprocal of the smallest quantity of a sample, which would give a positive coliform test. So just try to memorize this particular definition. This may be defined as the reciprocal of the smallest quantity of a sample, which would give a positive coliform test. However, with the advent of the membrane filter technique test, the actual number of coliform bacteria present in any given sample can be found, and hence the terms such as the most probable number or coliform index have become obsolete. So this is what is very important, since we are now talking about confirmatory test or membrane filtration technique, so where we can get exactly the number of coliform colonies present in the sample. From that perspective and from that context, it can be is said that the MPN and coliform index, both the approaches have become obsolete these days. Fine. So apart from that, we may have several other different type of impurities present in the water. So the first one is algae. Most of you might have seen algae. It's a type of plant that grows in water and flourishes in the presence of sunlight. Usually 7 types of algae have been distinguished by their color and shapes, and all of them contain green substances like chlorophyll, and that is why it is easily identified if it is present in the water. And this presence of chlorophyll permits them to utilize sunlight for their growth by conversion of carbon dioxide, water, and other simple substances into organic carbon compounds. And this is the reason, if the sunlight comes directly on the water bodies, algae flourishes in the sunlight and hence can be best controlled by covering the reservoir and so as to exclude the intrusion of sunlight from reaching into the water body where we can see that algae has already present. So there are different type of algae, but that is not required for your this particular course. So next is fungi. So fungi are those plants which grow without sunlight and live on other plants or animals or dead or alive. So some fungi, for example, toast tools may fairly be very large. So we can have different type of fungi, but mostly fungi are microscopic, and they may sometimes faced in water mains and may have to be removed with the chlorine treatment because they are considered as the impurities in case of fresh water. And then there is a third one, that is plankton. So plankton are the microscopic plants and animals life that either swim weakly or float in the water bodies and serve as a food for small sea creatures and fishes. In most of the cases, they are not injurious to health, but they affect the palatability of waters by secreting certain oils which give bad taste and odors to the waters. Moreover, these planktons, if they are present, they will contribute to the overall, aesthetic aspect of the water as well. So there might be higher TDS sometime, or you will see that the not TDS, the turbidity would be higher, and at the same time you can see that sometimes water is smelling differently, water is getting, the taste of the water is different than the usual. So those things can happen because of the presence of the plankton. Now, large quantities of plankton may impart green, brown, or yellow hue color in the water, so that is also very true. And they sometimes interfere with the smooth working of the rapid gravity filters by preventing the proper accumulation of alum or floc on sand. And if those kind of planktons are present in the water, then sometimes what it does is, if you see all those white porcelain utensils or porcelain's attachments in the kitchen and bathrooms, those will get yellow stains. So that happens because of those planktons and need to be cleaned or eradicated before you send that particular water in the distribution system. And this is the bacteriological quality of water. So the guideline is given, we can go through this. This particular portion has been taken out from the standard codal provisions. And if you see here, against different type of organisms, the guidelines in the right hand side has been given. So for all water intended for drinking, E. coli or thermotolerant coliform bacteria must not be detectable in any 100 ml sample. So that is the guideline should be maintained. Treated water entering the distribution system, so E. coli or any thermotolerant coliform bacteria must not be detectable in any 100 ml sample in this case as well. Total coliform bacteria also must not be detectable. Treated water in the distribution system E. coli or thermotolerant coliform bacteria must not be detectable in 100 ml sample, and you see for total coliform bacteria must not be detectable in 100 ml of sample. So you would be able to understand how serious this particular organisms or bacteriological aspects can become. And in case of large supplies where sufficient samples are examined, it must not be present in 95% of the samples taken throughout any 12-month period. So those are the things should be maintained or should be ensured before putting any water sample in the distribution system. So with this, we'll finish this particular chapter, and in the next chapter we'll be discussing about the let me we'll be discussing about the sources of the surface sources of different type of water. So sources and the storage of water, we'll be discussing these things here. And so we have finished the unit 1. Now we are going to talk about unit 2. So these units are mentioned in the syllabus. You can go through those units. Since waterborne diseases were there, and I discussed it before while talking about water quality, so I am not again repeating those things. We'll simply move to the next part. So that is source and storage of water. So if we try to see why water is so important to us, where in the planet where we are living, and we get three-fourths of the portion covered with water. So if you see, the ocean are the largest reservoirs of water, but the problem is it is saline, and that is why it is not easily readily usable for requirements of human survival. And from there the freshwater term has come, and if you see that 97.5% of the available water on this planet is saline, and which is why we actually have to focus on those remaining 2.5%. We are exploiting those 2.5% for all our purposes, and that's why we need to ensure sustainable utilization of those 2.5% water, since that is the only source of water for the next generation. So whatever we are planning, we are planning with those water, so we should ensure that that particular water does not lose its quality, and we should not contaminate them or pollute them in such extent that nature would not be able to decontaminate them by their usual process. And since we are unable to maintain these days because of the huge population and ignorance of the people, we are talking about decontamination processes. There are several eco-friendly decontamination processes which people are these days adopting to ensure that those 2.5% fresh water remain unaffected or unaltered in terms of its quality even after utilizations. So that is very important to remember over here. Now coming to the next slides, you will be able to understand, so among that 2.5%, how and how much is available to us for direct utilization. So among those 2.5%, 74% fresh waters are actually arrested in ice caps and glaciers. So you need to realize this part, that that means 74%, that means three-fourths of that 2.5% is not usable right now. They are in different physical state, and they are arrested in ice caps and glaciers. So now we'll have to see the rest of the 26%. Now among this 26%, 25.6% are groundwater, and though mostly the practice is to take out the groundwater, but it is actually cost extensive process. Now when we talk about just providing water for an entire city or entire municipality, it's not a small quantity of water which we need to deal. So that is why if we have surface sources like lakes, rivers, or bigger water bodies, so then in those cases it is much easier and economically viable. If we don't have any choice, obviously we'll have to go for those groundwater, and then we need to talk about those 25.6% of 2.5% which we have seen in the previous slide. Now, a global water content has been given over here. Now, another one thing I forgot to mention here, apart from lake and reservoirs, whatever soil moisture we see, like, within the soil matrix, some amount of water will be present in terms of, there are different form by which, water molecule can get attached with the soil molecules or soil particles, and that also contributes to those 0.4%. Now here I am not going to read out the table, but this table has been given over here just to demonstrate the overall global water content, and in terms of meter cube you can get an idea, and in the right hand side we have the percentage given, and the same thing was discussed in the previous two slides, so you can go through them, or you can have a very good idea about the overall volume which we consume, which we are unable to consume, which are available in different form in the nature. So those things can be understood very well from this particular table. So now, in the beginning of the source, whenever we talk about any source for that matter, one thing we need to mention is hydrologic cycle, because hydrologic cycle is one thing that can be said that it is the main source of water. This is the main source of water. Hydrologic cycle actually helps in Am I audible? Hello? Hello?
Yes, sir, you are audible.
Yes, sir.
Okay. So which was the last slide you could hear?
Hydrologic cycle.
Okay, so good. So hydrologic cycle, okay. So I'll start with that slide only. Just wait. Are you able to see my screen? My screen is visible. Hello?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yeah, so so we are talking about hydrologic cycle. I don't know how much you could hear when I was discussing. So entire process, most of you are aware of what happens in hydrologic cycle. What I wanted to mention is, one thing is very, very important. You will be able to remember, you'll be able to realize from here that, once this groundwater table is contaminated, it is really, really difficult to decontaminate them. And that is why, while dealing with industrial waste and leachate from landfills, and while managing them, we'll have to be more vigilant, so that those things cannot those things does not um, get mixed with the groundwater. Because if that happens, what will ultimately the result will be, among the 2.5%, whatever you know 24 or 25% we get as the fresh water, because rest among the 2.5%, maximum is 74% is arrested as the ice cap or glaciers. So rest, whatever are available, we are reducing the scope of utilization that water, because we are contaminating them. So we'll have to be very, very vigilant that those waters does not get contaminated. And these days, since the number of population is so high and so many other things are going on, people are less bothered about the environment, so they tend to ignore these things. That is why, now people are talking about decontamination of water and soil. So decontamination of water, so many people throughout the across the globe, you will see, that are conducting research to how to efficiently decontaminate these groundwater table, and as well as the surface water, so that the water which is getting contaminated because of over exploitation can again be utilized. And how can we minimize the cost of decontamination? So those things are the research aspect of this study. So I am not going to go in further details with hydrologic cycle, since these things are quite familiar to all of you, since you have this hydrologic cycle was discussed extensively during your ecology and environmental science course also. Now there are few definitions which should be always kept in our minds. So the first one is precipitation. So what is precipitation? Is the evaporated water from the surface of the streams, rivers, sea, ponds, wet surfaces, trees, plants, etc. It the entire process is basically written over here, again returned to the earth surface by the condensation in the form of rain, hail, dew, or sleet. And that is what is known as precipitation. So the major part of the precipitation occurs in the form of rain, and other forms of quantities are very small depending upon the prevailing weather conditions. Now the water of precipitation further goes off in the following ways. So the first one is runoff, second is infiltration. So the several components which we have seen in the hydrologic cycles are now being discussed over here. So after precipitation, a portion of its water flows over the ground in the form of rivers or streams, or in the forms of surface water streams. And some water flows towards the lakes and the ponds, and gets collected over there. So those are basically the form of runoff.
whereas infiltration is a portion of precipitation percolates in the ground and then it is stored in the form of subsoil or groundwater. So that is what is known as infiltration. So try to understand the difference of infiltration and runoff. Next is evaporation. That is also a very important part of the hydrologic cycle. So some portion of the precipitation is also evaporated from the lakes, reservoirs, rivers, or any superficial water bodies in the form of vapor due to the sun's heat is known as the evaporation. Now, this evaporation significantly contributes to the formation of cloud. Evapotranspiration that happens from the roots of the trees, basically what it does, it sucks up the water from the ground, from the soil moisture, and then some portion of that evaporates in the atmosphere through the leaves in the form of transpiration. So that is basically known as evapotranspiration. So these are the four important components which we usually discuss while talking about hydrologic cycle. So I wanted to discuss up to this point in today's class. Rest of the things we'll discuss in the next class.